Before any uptake of nutrients by plants can occur, nutrients must move to roots. Here, we'll cover the factors that control the movement of nutrients to roots. Roots gain access to nutrients by three mechanisms: diffusion, mass flow, and root interception. So in this section, we're going to cover the factors that control the diffusion of nutrients to roots, so the mass flow of nutrients, so the flow of nutrients along with water, and the amount of nutrients that are intercepted with roots as they grow through the soil. First, we'll start with diffusion, and diffusion delivers most nutrients to plant roots, meaning that most of the nutrients that are acquired by plant roots are delivered to roots via diffusion. So diffusion is the number one most important pathway by which nutrients are delivered to plant roots. Now, diffusion happens because there's a concentration gradient, and it's the relative concentrations of nutrients next to roots versus away from the roots that drives the diffusion of nutrients along that concentration gradient. So concentration is relatively high, concentration is relatively low, nutrients diffuse down that concentration gradient. Now, the concentration of nutrients in different portions of the soil, next to the root, away from the root, are dependent on the relative balance of mineralization, or the rate at which nutrients are made available in soil solution, to absorption. So next to the root, we need to know how much is being absorbed by the root, as well as how much is mineralizing and entering the soil solution next to and away from the root. If we have a lot of absorption that's happening next to the root, that concentration gradient is often going to look like this, and nutrients are going to diffuse downhill. Another factor that we need to think about for determining concentration and concentration gradients is the amount of buffering that happens in the soil. Soils that are highly buffered can supply nutrients to soil solution independent of mineralization by microbes, for example. And we need to think about cation exchange capacity. So if we're thinking about buffering and we have clays with a negative charge in there, having a lot of negative charge is going to hold on to a lot of cations. Probably a better way to draw it. So having a clay with a lot of cation exchange capacity, you tend to have a lot of cations that are associated with those clays. A large cation exchange capacity means a lot of buffering, such that when something like potassium in solution is absorbed by a root, then you can get exchange of potassium from clays into soil solution, assuming something like H+ is going to go and replace the potassium. So having a lot of buffering, having a lot of cation exchange capacity is something that can increase concentrations and maintain high concentrations away from the roots and maintain that concentration gradient. Now, the rates of diffusion, so independent of the concentration gradient. So if this is our root and we have a concentration gradient that looks something like this, let's say, so low concentrations next to the root, high concentrations away from the root, that concentration gradient is going to look like that. Now, the rate at which nutrients that are over here diffuse to over here along that concentration gradient is going to depend on a couple of factors. One of those is the charge density. So if we have a lot of clays with negative charges on them, for example, and the charge of the cation is positive, and let's say the root is over here, for this to get to the root along the concentration gradient and be taken up, it often interacts with clays. So it interacts with this negative charge, might be released, and then this negative charge, and then finally gets to the root. And that's going to slow down the movement of potassium from soil solution that's distant from the root to the root itself. Something like calcium, which has 2 positive, is +2, is going to be even more attracted, and that's probably going to be an even slower movement. In contrast, we have cation exchange capacity, so we have negative charges, we have our root. Something like nitrate, which has a negative charge to it, isn't going to interact with that clay at all, and it's going to zip straight over to the root. So charge density is important. In addition, the length of the diffusion path determines how fast nutrients are going to diffuse to the root. So let's say you have your root, and over here in solution you have your cation. If it's a really wet system and you have a continuous water film like that, potassium has a straight shot to get to the root. But if this is your root here, and this is potassium, and these are soil particles, for example, and that's the film that works along the soil particles, then potassium needs to work along this circuitous route. And that's going to take longer than if the soil was really wet and it had a straight shot. So the length of the diffusion path is important, and that length of the diffusion path is dependent on the particle size and the moisture level. So if you have a lot of big particles in your soil, for example, with a lot of water between them, it's a pretty much a straight shot. But if you have a lot of small particles and not a lot of water, you potentially can have a very circuitous route to get to the root. So large particles, a lot of water, faster diffusion. Smaller particles, not a lot of water, slower diffusion. Another factor to think about is that for plants, or at least roots that are taking up nutrients, that there's a zone around that root that it can access the nutrients. Nutrients can diffuse relatively fast, that zone is larger. If they don't diffuse very quickly, for example, that diffusion shell might be smaller. So we call these diffusion shells. It's a shell, it's a theoretical construct that essentially thinks about the concentration gradient and the distance over which roots can deplete nutrients. So this is what the nutrient might be in the bulk soil, and this is what the concentration gradient is till you get to the root surface. So we think of these as shells around roots of depleted low concentration. So if you have a root in the soil, there might be different shells of depletion around it depending on the nutrients that it's taking up. If we take a look at the concentration gradient of something that diffuses relatively well, and that concentration gradient might look like that. If it doesn't diffuse well, or it actually isn't being taken up by the root, there might be no change in concentration as you go with this away from the root. Now that's a little bit hard to see. I've actually worked on this question directly, and I had put together a model which we call the spatially explicit representation of the movement and uptake of nutrients in order to see this a little better. And the model was really simple. It just divided the soil into a grid and allowed there to be diffusion between individual grid cells depending on what the concentration gradients were. So high concentrations in this cell of a given nutrient, low concentrations here. At each time step you'd have diffusion from high to low. We'd also have roots that would occupy some of those cells, and they would take up nutrients based on what the concentrations were in the external or adjacent cells. If you do that and you have a lot of grid cells, you can, for a reasonable volume of soil, so here this is 2 centimeters on a side, on both sides, take a look at what these diffusion shells look like. So there's a lot of grid points in here and you can't see them, but each of these dark blue spots represents a root. And the concentration gradient here goes from yellow would be high, or red would be high, blue would be low. And you can see how for the element that we were working on, you get these shells of diffusion or shells of depletion around individual roots. And where there's clusters of roots, you have larger zones of depletion. In areas where you don't have as many roots, it's going to be more open. So this is a good picture to keep in mind of the concentration gradients. This again is a two-dimensional slice of the soil where you have lots of roots, and this is a pretty typical root density for a grassland that we would have there. And you've got areas of relatively high concentrations and very low concentrations around the roots. Now, for plants, once diffusion shells are generated, how do plants increase the nutrient supplies? Well, one option is to increase the proportion of soil that can be exploited. So, if we think of this as a two-dimensional slice of soil and roots grow through that soil, and let's say they branch off, you can have a depletion zone around here of lower concentrations and high concentrations adjacent to the root, but outside of those depletion shells. One way to increase the nutrient supply is to produce new roots in these areas and then deplete the zone around those roots. So that's one way you can do that. But on an ecosystem level, at some point, there's not going to be an area of the soil or volume of the soil where there are no unexploited soils. Everything is going to be within a diffusion shell. At that point, all plants can do is really compete for nutrients. So you have your soil, you have roots that are growing through the soil like this, and there's going to be another species that has its roots like this, and there's really very little area that we can consider outside of the diffusion shells of these individual roots. And then all those plants can do is essentially compete for the nutrients that get made available in the soil somewhere and diffuse along gradients to roots. Now, plants can increase in supplies of nutrients through different factors that we'll talk about later in different chapters. But essentially, if you think of supply as being external to the plant, all they can do is exploit as much of the soil as they can and then just wait for nutrients to diffuse to those roots. So that was diffusion, and that was the number one factor that supplies nutrients to roots. The number two factor, that second factor, is mass flow. Mass flow is the movement of dissolved nutrients to the root surface in flowing soil water. So you can have a root that's in soil, and you can have a concentration gradient that looks like this, and that generates a diffusion shell around that root. But water can flow through the soil. So water itself can move along different pathways and deliver nutrients into diffusion shells. So to understand mass flow, we simply have to ask, why does water flow? Well, for example, we have transpiration. So transpiration, as you remember, if we have a root, they're going to be taking up nutrients, but they're also going to be taking up water. And that's going to cause water to move towards the roots and bring along nutrients with it. So although this has the potential to deliver nutrients to roots, usually diffusion is more important than mass flow. And essentially the reason for that is that if you have a concentration gradient, the diffusion of nutrients along those concentration gradients is usually a lot faster than the movement of water to the root. And so even though this has the potential to move nutrients to plants, diffusion happens just a lot faster. And by the time this water gets here, those nutrients would have diffused long ahead of it. Now there are situations where you can have saturated flow of water. So if you have a soil surface, you have roots in the soil, and it rains a lot, you're going to get downward movement of water. And that actually can be relatively fast. And when that happens, it's often fast enough to replenish diffusion shells. Depending on your root density, though, all this might do is take a situation where you have a concentration gradient and equalize it out. But still, that's a delivery of nutrients to the root. And I'll actually try and draw that better. So here is a distance from the root, and this is going to be the concentration of an element, and we'll just say nitrogen. If this is the root surface right here, you're going to have a concentration gradient that looks like that. But if you have a wetting front that happens, so water starts to move through the system, that concentration gradient is going to become flat as nutrients are equalized out. And that's actually a delivery of nutrients, increase in the concentration right at that root surface. So sometimes you can have mass flow that is fast enough to replenish diffusion shells, but that's a relatively rare event compared to the importance of diffusion. Now the last factor that we talked about in addition to diffusion and mass flow is root interception. And what root interception is that if you take a look at the volume of soil and there's going to be nutrients in it, that the movement of roots through that soil, just as they grow, is going to pick up nutrients that are present in the volume of soil that it displaces. So you have a root, and that has to push often away soil as it moves through the system or work its way through the soil in between particles. And as it does that, it's going to intercept nutrients at its tip. But it's actually not a lot of nutrients, and it's not important. So of the three, number one is diffusion. Number two is mass flow. And so really understanding diffusion is most important, and under the conditions for which mass flow can be important. Interception, generally not that important, and it's important to understand why. Essentially, the amount of soil volume that it displaces and can directly intercept nutrients, it's just too low, and there's not a lot of nutrients there. So it's not really a big factor.